Welcome to the Environmental Genetics Laboratory. We are situated in the campus of University of Latvia. We are the team of 11 scientists covering different fields of environmental genetics and biotechnology. Our main research focus right now is estimating influence of electromagnetic field on test organisms. And now I would like to invite you to join video tour to see the process of our research methods.
Our laboratory performs research with various in vitro plant cultures. They are whole plants or parts of them, like organs, tissues, or cells, grown under sterile conditions in cultivation vessels. In vitro cultures are a suitable model for studying the effects of various factors because the side effects on plant growth and development are eliminated. The obtained results are relatively fast. All operations using in vitro cultures, maintenance of plants collection, preparation of experimental sample sets, and so on, are performed in a laminar box, which provides sterile conditions. To determine the effects of the electromagnetic field, as the model organisms, we are using duckweed and tobacco plants, as well as different plants tissue cultures. During the experiment, we want to find out if an electromagnetic field has a biological effect on living organisms. In the electromagnetic field's generation, a Helmholtz coil is used. It generates a constant magnetic field till 700 microtesla. The experimental material is placed in the center of the coil for a determined period of time, in order to analyze if there are any changes of development, lifespan, or morphology. We compare exposed organisms to control non-affected groups. Fruit flies are used as test objects during studies of the influence of electromagnetic fields on living organisms. There are two experiments going on at the same time. One that contains 20 lines of flies obtained from a batch of 50 eggs, another one obtained from 2 female flies. 10 lines from each experiment are irradiated in an electromagnetic field using a Helmholtz coil, which generates a constant electromagnetic field. The other 10 lines are placed in a safe environment as a control group, which lives under normal conditions. After a certain period of time, the test objects are checked by performing a speed test, during which the flies travel a certain distance and are timed, later calculating their movement speed, counted and checked for mutations. Using flow cytometry and cell sorting, it is possible to determine more than 20 different cell parameters. Flow cytometry methods are widely used for detection of changes of cell psychological state. Flow cytometry method allows to evaluate a large number of cells in a very short time, and the obtained results are statistically significant and representative of the whole population. For the analysis, we use a rather unique BD-FACS JAS cell sorter. In flow cytometry, the cell suspension flowing through the capillary is analyzed by light scattering or fluorescent signals after the cell is initiated using a laser. For flow cytometry, it is possible to use cells, cell nucleus, and smaller objects that are smaller than 50 micrometers in diameter. For that reason, preparation of sample plays a big role. For example, a plant leaf part is mechanically chopped with a razor blade in a lysis buffer and is filtered. In such a way, individual cells are obtained, which are later on used in experiments. For studying the influences of different environmental factors, gametic cells are the perfect model. Gametic cells are very sensitive to different factor influences, and if plant gametic cells are used in studies, they are quickly obtainable in large quantities. Plants' buds are chopped in the blender. With a centrifuge, the live pollen is separated. 1 milliliter of cell sediment contains about 600,000 cells. The obtained pollen culture is influenced by the electromagnetic field. By staining the cell compounds of interest with a fluorescent dye, it is possible to enumerate the cells, determine the changes in fluorescent caused by various stressors, determine the relative amount of DNA in the cell nuclei, and even differentiate the various cell types in suspension. Sample preparation is based on the selection of the required fluorescent cell compounds and fluorescent mode. In studies with living cells, changes in cell fluorescence are determined. To estimate the relative amount of DNA in the cells, we chop the tissue material in the extraction buffer, we stain the cells with propodium iodide, a dye that binds directly to DNA. When performing genetic researches with molecular methods, DNA of the model organism is analyzed. Therefore, the precondition for successful research is the acquisition of pure, unadulterated, and unfragmented DNA. DNA is a molecule that stores information about the heredity of a cell, determines the structure of the cell, the set of biological and psychological properties throughout the life cycle of the cell. Genetic diversity studies analyze changes in the DNA sequence of an individual's under the influence of various biotic or abiotic factors, while population genetics studies differences in DNA between individuals. There are several basic steps in DNA extraction, the content of which may vary slightly depending on the species being studied. Initially, cell membranes are destroyed both mechanically and with help of extraction buffers. The various saline solutions, bounded by cell lipids and proteins, are separated and removed by centrifugation. Depending on the species, DNA extraction may require specific additional steps to remove, for example, phenols or other aromatic compounds. Finally, the extracted DNA is washed out from impurities using various saline solutions, chloroform, and various alcohols. When DNA is extracted and purified, in order to make further analysis possible, it is necessary to multiply DNA fragment or sequence of interest. The DNA fragment which is being analyzed is multiplied thousands of times. That is done during the polymerase chain reactions. All reagents, after they have been mixed together, are placed in a thermocycler, where DNA is replicated during multiple cycles. Replicated DNA fragments are analyzed by gel electrophoresis. On the gel, the DNA fragments are separated depending on their size. Shorter fragments move faster and go further on the gel. After electrophoresis, the DNA fragments are stained with ethidium bromide, and finally fragments become well visible using ultraviolet light. At first, DNA sequences of interest amplified by polymerase chain reactions are analyzed using high-resolution capillary gel electrophoresis, in which charged DNA molecules move in the gel under the influence of an electric field. DNA fragments of different lengths are separated from each other based on different mobility of these fragments. The shortest fragments migrate faster in the gel, the longer ones slower. Fragments are identified using the fluorescent dye added to DNA sample by recording the fluorescence intensity. In genetic diversity studies, the specific DNA fragment to be analyzed is determined by the fluorescence intensity peaks. It is or is not present in the sample. In population genetics, the DNA fragments of interest are viewed and analyzed for many individuals altogether. Mutation detection at DNA level is carried out by our collaborators from the Scientific Research Institute Nature Research Center Laboratory of Molecular Ecology. They used catalyst glutathione peroxidase and other gene sequencing in order to detect changes of DNA strand caused by stress factors, including EMF, examining its influences on different model objects. Now we are located at the Microscopy Flow Laboratory, which is equipped with microscopes and computers which are interconnected, and together there are 25 workstations. We study cell cytoplasm flow of Canadian waterweed in a microscope with and without the influence of a mobile phone. In the moment of switching on the phone, a possible change in the chloroplast movement is detectable. The reason is that a mobile phone causes stress for the cells. We use live cells because the Canadian waterweed plant is a forgiving test subject. It is relatively easy to see through because the cells are located in 2 layers.
I hope this inspires you for new research ideas that we can pursue together. Feel free to contact or visit us.